Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Simon Aberg, Senior Research Engineer from Cytiva.
Hello, everyone, and welcome to today's webinar with the title A Scalable Single-Use Two-Step Plasmid Purification Process, hosted by me, Simon Aberg, a Senior Research Engineer in the Viral Vector Applications Group within Genomic Medicine R&D at Cytiva. So we'd just like to start with some contents. We'll touch a bit about the background of plasmids, talk a bit about the aim of the project, then go on to show the actual results, then talk a little bit about the analytics and the criteria for regulatory acceptance, and then finally we'll do a summary and conclusion. Okay, so let's talk a bit about background about plasmids. Well, as you probably know, plasmids are small DNA molecules that are physically separated from chromosomal DNA. They are circular and double-stranded that replicate independently, and as such, they are a very important genetic engineering tool used to clone and amplify or express genes in molecular biology. And as of now, they are completely essentials for the production of viral vectors and mRNA. And we like to think of that plasmid simplified exists in different topologies. And simply, we usually describe those isoforms as the linear one, the open circular one that we think of as a normal hair tie, and then the supercoiled one as a supertwined hair tie. And within bioprocessing, it's the supercoiled isoform that we want to enrich. And within bioprocessing, we usually work with plasmids in the range of 5 to 20 kilobase pairs. Okay, let's move on and talk a bit about applications. So plasmids, they are the start for many therapies. First, they are used as template for mRNA manufacturing. They are used in recombinant protein production, or as is for DNA vaccines, and for manufacturing of viral vectors. And the scale here and quantity of plasmids, it will depend on the application area. So in this slide, you can think of as on the left-hand side, you have the largest scale, and then gradually as we move on to the right, the scale required decreases. So first we have DNA vaccines, where plasmid DNA is used directly as a therapy or a vaccine. And the quantity or scale of plasmid here is typically small or medium to very large. It will depend on the intended target population. This technology is still evolving and hasn't seen great success. It's been tested several times in clinical trials, and so far the only approval we've seen is the Indian emergency approval of SARS-CoV-2 COVID vaccine. And the issue is here that when you deliver plasmid as a therapeutic, you need to deliver it to the nucleus, which means that it first needs to be delivered to the cytosol and then into nucleus, which have shown to be a challenge. And then we have viral vectors, multi-plasmid systems used for transfection in each viral vector batch, lentiviruses, AAVs, etc. The quantity or scale here is usually medium to very large. It will depend on the vector and therapy, but for instance, for AAV, For transfection, you usually need a system of 3 to 4 plasmids for the transgene, the RepCap viral genome, and the helper plasmid. So usually you need quite a lot of plasmid. And this technology is still evolving. We are seeing a lot of candidates in trials and probably will just see more coming. And then we have mRNA. We need one template for each back, and the scale here is not usually that big. Actually, we can support a 50-liter fermentation. We can support around 100 million doses. So you actually don't need that much plasmid here thanks to the IVT reaction. Also a technology that is evolving a lot, seeing great success in recent years with the 2 approved COVID vaccines. And then we have a protein where pDNA is used for the initial cell line and cell bank development. The scale is usually quite small and not necessarily GMP, and it's an established technology with many approved therapies. Okay, let's start to go into the aim of this project. So the aim of this project was to address some of the challenges of Cytiva legacy plasmid DNA process offering, as you see in this flowchart here. First, we have a conventional fermentation, lysis, and clarification steps, then followed by a 3-step downstream process, starting with a size exclusion chromatography step at first, followed by removal of open circular plasmid DNA using Plasmid Select Extra, and then a final polishing step using an anion exchanger. The advantages of this process was that it was robust. It did fulfill the regulatory requirements. However, it had some challenges. It's a 3-step process, which means it's quite time consuming. It had some scalability challenges. Not all steps, namely the first size exclusion step, was fully scalable. Neither were all columns single-use compatible and compatible with the Cytiva ready-to-process formats. And then lastly, it consumed large amounts of ammonium sulfate. So what we wanted to address and the process characteristics we wanted to improve with the new process was to achieve clinical grade material, have a scalable single-use compatible process, reduce the process time, and have a 2-step purification process that would also be able to reduce the amount of ammonium sulfate used. Then we will move on. So, and what would be the acceptance criteria for success for this process? Well, we had as a target the US FDA regulatory guidelines for plasmid DNA. And then we had a bit more stringent requirements for redundancy. For instance, the guideline says 80% supercoil, but we know now the current GMP is 95 or better. And then I think we can move on to the results. And before we go to the downstream part, I just want to shed some light on the upstream and clarification processing, because it is a very important part. And you need to consider the whole process. So we think that optimizing the upstream and clarification part can really give you advantages later on downstream. For instance, the open circular supercoil ratio can be greatly influenced by your fermentation. You should take care to have, for instance, excessive addition of antifoam, and also to optimize your feed profile to avoid glycerol accumulation that we also seen can negatively influence your open circular supercoil ratio. And then there are different options for cell concentration. In large scale, you can use microfiltration via TFF or single-use continuous centrifugation. And in lab scale, you can use normal centrifugation. And then for the lysis, parameters here are critical to maximizing the pDNA yield, because the purpose of the lysis is to release the plasmid DNA while not doing it too harsh to avoid create excessive shear of genomic DNA, host cell proteins, RNA, or endotoxins that might later on will be hard to remove. And we think that one great way to do this is to reduce the amount of RNA by calcium chloride precipitation directly after lysis. It's a fairly easy, fast and cheap way to greatly reduce the amount of RNA. So the message here is to set up your downstream process for success by optimizing the upstream and clarification process. But now I think we can move on to the downstream part. So the new plasmid DNA process that we developed is outlined here. I will just mention that there are different processes, and Cytiva with our single-use equipment offering will support the different processes of your need. But let's look a bit more on the downstream part. Okay, so here you have the new developed downstream process on the top and the old legacy process on the bottom. So we developed a new process where we replaced the first Cepharo 6 fast flow size exclusion step with capture using Mustang Q XT membrane adsorber. And then we replaced the second Plasmid Select Extra step by our updated resin capital Plasmid Select instead. And by doing so, we were able to increase the capacity, improve the pressure flow properties, but also remove the last polishing step, the source 30Q. And now I will show you some data on how the first Mustang Q capture first can look like. So here we have capture using Mustang Q membrane adsorber. On the left, outlined in blue, you can see the lab scale using the Mustang Q XT 5 milliliter capsule, and to the right in green, you can see the production scale Mustang Q XT 140 milliliter capsule. The loads here were for the lab scale 12 milligrams per membrane volume, and for the production scale 9 milligrams per membrane volume. But we've seen a capacity of up to 16 milligrams per plasmid DNA per membrane volume for this plasmid. But of course, the capsules come in discrete sizes, so depending on your load will differ depending on how much material you have. And as you see here, the flow rate we've used is 5 membrane volumes per minute. So for the small capsule, that equals 25 milliliters per minute. And for the larger capsule, that equals 700 milliliter per minute. So it's very high throughput. Then we eluted the plasmid, as you can see here, using 3 molar sodium chloride. You can see that the yield for the lab scale was 58%, and for the production scale, 67%. And I want to mention here that plasmid DNA binds very strongly to anion exchangers, so high yields can be difficult. Maybe if you really want to push the yield, you would have to implement multiple elution cycles to really get as much material out as possible. But let's look at some data on the capital Plasmid Select step. So here we have the enrichment of supercold plasmid DNA using capital Plasmid Select. It's the similar view as on the last slide. You have the lab scale using the 18 milliliter column on the left, and then the production scale where we used a 1 liter ready to process prepared column on the right. Here, the loads were 2.8 milligrams per milliliter for the lab scale and 0.9 milligrams per milliliter for the production scale. And we've determined a capacity of up to 3 milligrams of plasmid DNA per milliliter of resin for this one. And then you can see that we eluted the plasmid using 11 column volumes against water. You can see there that the open circular isoform elutes first as a very small peak, and then the supercoiled isoform as the larger peak. You can also use a isocratic elution here, but that would require some optimization of the buffer conductivity, because the separation is very intricate, so the exact conductivity would depend on your plasmid and the sequence. The step yield here was 77% for the lab scale and 72% for the production scale, which is quite typical yields for this kind of step. Let's move on and look a bit on the total process yields. You can see the step yields there, that we had quite high step yields in all steps. The lowest of step yields we had for the Mustang Q step. So the yield of this step will really, to a large extent, decide how big total yield you get out of your process. And then let's move on and look to some other benefits of the process. Here you can see on the left a comparison of process time in hours for each of the unit operations between the legacy process in green and the new process in blue. And then you can see by going from 2 unit operations to 3, and also replacing the size exclusion column with the Mustang Q, we could reduce the process time with 60% for downstream processing. And then on the right, you can see a comparison of the required amount of 2 molar ammonium sulfate buffer that you would need for downstream processing between the legacy process in green and the new process in blue. And when you look at this one, it becomes evident that the first Zephyr 06 fast flow size exclusion step consumed large amounts of buffer, and only by replacing that one we can save large amounts of ammonium sulfate buffer consumption. But let's move on and look at some of the analytics and the criteria for regulatory acceptance. So we can conclude that the production scale pDNA process fulfills the FDA regulatory requirements. You can see the Cytiva and the FDA acceptance criteria in bold black in the bottom. And you can see that we actually already fulfilled all the regulatory requirements after the Mustang Q. But this large scale was a single run process with a very successful fermentation, and that is not necessarily always the case. So we really think that the additional plasmid select step is very important for redundancy of your process, to be sure that you can achieve your quality attributes. Let's move on and look at an agarose gel showing the reduction of RNA using calcium chloride. So here you can see a gel with each unit operation loaded in each well. And then you can see in the bottom, the white smear, the reduction of RNA over each process step. And actually you can see that already after the lyset, we removed most of the RNA. So yes, calcium chloride precipitation is a very effective and simple way to facilitate easier downstream processing. And then I also want to show for analytic some chromatographical methods for plasmid DNA analysis. So for this project, we've been using our own in-house Zephyros highest performance method for accurate plasmid DNA concentration determination. And here to the left, you can see a comparison of our method in blue with a microvolume spectrophotometer in green. And then you can see the yellow line that represents the 8:60, 8:80 ratio. So how much RNA we have left in the sample. And what you can observe here is that at when we still have some residual RNA left, the spectrophotometer tends to overestimate the amount of plasmid DNA that we have in the method. But since our method first separates the two and then we can determine the concentration, we can get an accurate pDNA concentration independent of sample. Not only that, we can also determine concentration in all type of matrices, for instance, in early lysate pools or etc. And then to the right, we show data using open circular supercoil determination using the analytical capital plasmid select method, which is also a very quick and easy method. Takes only 15 minutes for one run, and you can quickly determine your open circular supercoil ratio. Here we compared our method to state-of-the-art capillary gel electrophoresis, and we can see that our method is comparable and performs equally. So both of these methods are very convenient and fast methods that you can set up on a Pure 25 system in your lab. They are great tools for your process development needs. And I think with that, I want to move on to some summary and conclusions. And I want to summarize by saying that the two-step plasmid process, it is scalable and it fulfills all the regulatory requirements independent of your plasmid application. And we show rapid capture of plasmid DNA using Mustang Q, followed by selective purification of supercoiled plasmid DNA using capital plasmid select. And compared with our old legacy three-step process, we have a 60% reduction in downstream processing. And we show efficient RNA reduction using calcium chloride precipitation with improved sustainability by a 90% reduction in ammonium sulfate consumption. So with that, I just want to show some acknowledgement and thank all my colleagues who've supported this work. And with that, I want to thank all of you who's been listening, and I welcome any questions or comments that you might have. Thank you so much.
Okay, great. Thank you, Simon. And as a reminder to our audience, you can go ahead and type in your questions for Simon now. So our first question is, why enrich the supercoiled isoform of plasmid?
Yeah, so that would be that because in the open circular and the linear isoforms, they're usually formed sometimes due to the presence of nicks in the DNA strand. So therefore, enriching only the supercoiled isoform is a means of quality assurance to avoid plasmids with potential mutations.
All right, and our next question is, what is the homogeneity requirement of pDNA for mRNA vaccine production?
Now, so this has been a bit of a, or it is an ongoing discussion, because as far as I'm concerned, I don't think there is any FDA guidance yet of pDNA as an intermediate, as for instance for mRNA or for AAV, etc. Therefore, we don't really know. I guess it's something that will develop as more and more drugs pass their BLA and get accepted, and maybe we'll get some more guidance there. But right now, I think the requirement is still 95% supercoiled, and probably I would say it's continue be 95% supercoiled plasmid DNA.
And does
so the same as for a clinical application. Oh, sorry.
No, I'm sorry. Do you have anything else you want to add?
No, I think that was
okay.
That was the answer.
So our next question is, does this new flow of scheme eliminate buffer exchange or TFF step on lysate prior to chromatography?
No, I don't know if you missed, but in the flow scheme, yeah, I don't know, can I click here and show? But we do have a TFF step prior to the chromatography. And then partially is to get the correct binding buffer for the chromatography. And also, as we showed in when we displayed the analysis of the quality attributes, that you actually do remove quite a lot of contaminants, mainly RNA, during the TFF. But there is potential to load more crude lysate, but you would need to optimize the conditions. And I think if you, like we did, employ calcium chloride precipitation, you will have a very high conductivity in your sample, and that would make it very difficult to bind the pDNA lysate crude to the Mustang Q without a TFS step.
And how do you measure scDNA concentration?
Yeah, so that I think maybe this question came in before I covered it on slide 21, but yeah. So we've used our in-house analytical version of our capital plasmid select resin in the high trap format, which we think is very convenient and easy to use, and which we also showed that is comparable to state-of-the-art methods of capillary gel electrophoresis. So that is the method we use, and as I said, it's a quick and fast method that only takes around 15 minutes per sample.
All right, and our next question is, thank you for an interesting presentation. I have a question about hands-on time. Have you showed that the process time is lower, but what about worker hours in regards to preparation and hands-on purification times for the old and new process?
Yeah, so it would depend, of course, if you have one less unit operation to set up, you will have probably have reduced the process time there as well. But of course, you need to make buffers, even if you replace with a faster step. But we haven't made that exact analysis, but we think that we're reducing the amount of unit operations. You would also, in some way, reduce the worker hours in preparation as well. I think that is the answer I could give there.
And what is the column size for Captoplasmid Select analytical method?
So we've been using, it's a 1 milliliter high trap column, and we load about, well, it depends on the amount of sample, but for the method we load a 500 microliter sample on that.
All right, fantastic. Well, it looks like we're running just a bit short on time, so if we don't get to your questions, we'll be passing them along to Simon, who can follow up with you directly. So please go ahead and type in any questions you might still have, and we'll get those passed along. The last question for the webcast is, can the linear gradient be changed to isocratic in Captoplasmid?
Yes, it can. I think I also touched upon it during the webcast. It's possible. You need to optimize the conductivity for your specific plasmid, but essentially it is possible to pass the open circular isoform in the flow through, and then with an isocratic elution only elute the supercoiled isoform, as long as you can find a good big enough separation window in conductivity between your open circular and supercoiled isoform. But it's definitely possible, yes.
All right, fantastic. Well, thank you so much, Simon.
Thank you.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Thank you.